Hello, my name is Lakshay Anand. I am a bioinformatician currently doing PhD at the University of Kentucky EEG group headed by Dr. Carlos Lopez. Today, I am presenting a bioinformatics tool, chromoMap, that I developed as a part of my thesis, which is an R tool that can be used to create interactive genomic visualization of multiomics data. Basically, chromoMap is an R package written in R, JavaScript, and D3 library. Using the tool, users can map genomic features onto genomic regions. Users can construct interactive plots such that when the users hover over the region, the tooltip shows more information. Users can also visualize numeric or categorical data associated with genomic features. chromoMap renders chromosome as contiguous loci where a locus is a genomic range and can map multiple features within it, which can be viewed on tooltips. In contrast to this point annotation function, chromoMap also allows annotation of long genomic features as segments. To create the visualization, you need to provide two input files, one specifying the chromosome coordinates and the other specifying the annotation coordinates. The polyploidy function of chromoMap allows users to add multiple sets of chromosomes. This functionality could be useful, for instance, while comparing homologous chromosome or even for cross-species comparison. Numeric data can be visualized as heat maps. When the user hovers over a locus, it will show a detailed heat map for all annotations in that locus. Also, the value at a locus is determined by an aggregate function that users can change. Numeric data can also be visualized as bars or scatterplot. Users can also apply conditioned filtering to these bars and scatterplot. This is an example of coronavirus patients' gene expression data visualized using chromoMap. It is built with several functions offered by chromoMap. Let's take a moment to understand what the plot is showing. The first track visualizes the gene density. The second track visualizes the fold chain for the differentially expressed genes between COVID-19 patients and healthy individuals, where the significant genes are colored in orange. The third and fourth tracks compare the normalized gene expression between a severe and asymptomatic patient. The last track visualizes the differentially expressed gene, where the genes are color-coded based on their metabolic pathways. Here is another example of how epigenomics data can be visualized using this tool. This example visualizes the splice variants of human gene. It demonstrates the segment annotation functionality and also how the users can visualize categorical data. A special function of chromoMap allows linking of two genomic features present either within the same chromosome or on different chromosomes. Users can also associate and visualize numeric and categorical data related to these links. Links between segments can be visualized as chords. Here is an example where the function can be useful. Again, this plot also uses other functions offered by chromoMap. Here the expression of the orthologous genes in rat and mouse are compared where the genes are connected with links. I will now briefly go through some other functions of chromoMap. You can tag loci using the epi tag function. You can highlight a genomic region and further zoom in on it. You can add labels to the plot. You can also add hyperlinks to the annotations. chromoMap provides several customization options to create desirable plots. You can either use chromoMap as command line tool or as containerized web app that provide GUI access to its functions. You can export your plot as a PNG or SVG. To include them in publication, you can also include them in R Markdown or Shiny web apps. Or even better, use the plots as shareable standalone HTML documents that can be included in supplementary materials in publication. This is how people have used chromoMap so far. Thank you for attending. For more information, please scan the QR code.